Okay, guys. Everyone, thank you for coming. It's that time of the semester, I hear you. Okay. So this is hopefully the last lecture on skeleton we will see. Obviously, exam theories this week. I had a whole day of oral exams yesterday. Everyone was very excited to talk to me about the cytoskeleton. I know, I figure, yes, it's true. I didn't talk to you. You may as well come talk to me. So that'll be finished at the end of today.
You still have more?
Huh? No.
Oh.
No, the exam, examining. I'll be finished examining you people at the end of the day. Okay, so here's our take 5 minutes to write. Okay. Okay, guys. Muscle contraction is initiated by the release of calcium from the sarcoplasmic reticulum. What is the calcium doing at the molecular level to induce muscle contraction? Okay, how are we doing? Everyone good? Okay. Linda, I saw you. So tell me, how did you approach this? What do you think? Okay, so the calcium is causing the tryptomyosin to move out of the way, allowing actin heads to be available for binding by myosin. So specifically, though, what is the calcium doing? So you told me the outcome, which is correct. I want one more detail. Yes. Yeah. So it binds to the troponin complex, and that is what is holding that tropomyosin in place. And so once it binds to that complex, it releases the tropomyosin. So good. Anyone want to add to that? So that was an easy one. I figure on an exam today it would make it easy. But good. Okay, so far, most of this is what's on the exam. And the exam kind of stops at motor proteins. Then we talked on Tuesday a little bit about motor proteins and how they're regulated and about muscle contraction. So let's talk a little bit about other structures that are made, other cellular structures that are formed from by cytoskeleton. Well, they're all formed by cytoskeleton, but we'll talk about a few examples. Cilia and flagella, right? So these are motility structures, and they're built from microtubules and dynein, right? Flagella are in sperm and protozoa, and then required for swimming. And cilia, this is this wave-like movement of cilia in your, for example, in your tissues, like in your gut or ovaries, or like on a paramecium, which allows paramecium to move through fluid, or the cilia also allow fluid to move your eggs through your oviduct, mucus in the respiratory tract, all that stuff. And so the core, also called an axoneme of cilia and flagella, they're made of microtubule doublets. So these green, those are your microtubules, and they're doublets because there are two of them joined together. And they're arranged in a ring around a central microtubule doublet. And they're also bound together by these proteins here called nexin. So they're fixed in a way. And then there are also dynein. So you've got dynein motor protein and microtubules, and their organization allows the movement of the cilia or flagella. And I'll explain that. Right, so you have got this ciliary or axonemal dynein, and what it does is it binds to neighboring microtubules. So we've got these microtubules that are in doublets, and they're arranged around each other, arranged in a circle. And so you've got that ATP hydrolysis that allows movement of your motor protein, and it's going to dynein, so it's going to walk to the minus end of one microtubule while remaining fixed and bound to the other microtubule. Right? So normally you can imagine if you've got two microtubules, will be my arms right here, and the motor protein is the cargo cargo part is one microtubule, and it's walking along the other microtubule. You'll have sliding, right? But it's those contacts with these proteins that kind of keep them from actually sliding, so they're fixed. And really, what happens is they bend. So that's how you've got that bending. So you get this dynein movement along one microtubule. So you can imagine, like, here's your cargo part, here's your wheels, right? Walking along the track. So you get the dynein movement along one microtubule while remaining fixed to the other one. And that is what causes the bending. If you don't have these linking proteins, then you do just get sliding. So it's those linking proteins, like the bending, that's associated with flagella cilia movement. So if you have a defect in ciliary dynin, so a defect in this motor protein, what would be the outcome? Right, so there's actually a syndrome called Carter-Jenner's, I hope I'm not killing the pronunciation, syndrome, and that's where you have missing dynin arms. So this would be normal cilia, just like that picture I just showed you. And then this would be if you're missing your dynin arms. And so what do you think the symptoms, or what would the patient look like who has this syndrome? I mean, not what they would look like. It might just look like you and me. Yeah.
In the mucosal layer, the cilia won't be active, so you won't have the cilia pushing down possible pathogens.
Right.
So they'll be more prone to
Right.
sicknesses.
Right. So the cilia in their respiratory tract would not be able to move mucus, so you might not be able to get rid of pathogens, might be more prone to a lot of mucus and infections. What else? Yes?
Or digestion.
More indigestion, maybe. What else? Yep.
You said cilia is in the reproductive tract,
Yep.
so that won't be functioning.
Right, so cilia are in the reproductive tract and are allowing fluid to move down the aqueduct, so that could have an issue, could cause trouble with ovulation, right? Maybe not ovulation, but the egg getting to where it needs to go. What else? There's another one, cilia and flagella. What has flagella? Sperm. Yeah. What about your paramecium? Will your paramecium? Sorry, I couldn't even follow through with that goofiness. Okay. Where is your paramecium located? Where did you learn about that? In AMP. Did you guys learn where your paramecium is?
Isn't that a bad thing?
It's another organism. Yes. Everyone's just staring at me, like racking their brains, like, I know what Staffman is. Now you're asking me about my paramecium. It's just funny because of the way the book talks about it. It's like paramecium, and then everything else is part of an organism, like one organism. Anyways, sorry, just having fun with you guys. Right. So I guess this guy realized when he saw patients who had this kind of triad of symptoms that maybe they had an issue with their ciliary dynin, because they would present with all like with infertility and excess mucus and more infections and different kinds of symptoms that were all related to one defect. Okay. So some cytoskeletal structures are stable structures, right? Like cilia, right? Are they constantly being rebuilt and torn down? No, they're there. They're stable, right? And so you can probably make certain assumptions about the microtubules that make up cilia and flagella, right? They probably have limited dynamic instability, right? Thank you. Right. So, but other structures need to be constructed when they are needed, right? In response to internal or external signals. What kind of structures need to be constructed when they are needed? Mitotic spindle. That's like the one that should just jump right to your brain. Absolutely. Mitotic spindle. What else? Actin. Well, so actin, like the lamellipodia and filopodia that are associated with movement, those are structures that are made from actin, but only when needed, because the cell's not just readily moved. Well, maybe it is readily moving, but it usually moves in response to a signal. Right. So your microtubules and motor proteins are required for construction of the mitotic spindle, only when needed. Right. Not an interphase. I realize that a lot of people don't realize the interphase is all of the cell cycle minus mitosis, right? So just remember that. Sometimes people, I think, confuse it because it's something phased, I think they think it's a part of mitosis, but it's actually the rest of the cell cycle. But just to keep that in mind. So microtubules and motor proteins are required for construction of the mitotic spindle, right? They need to be these interphase microtubules that you see on your left. They need to be disassembled, and then they need to be reassembled into bipolar mitotic spindles. Apparently, this happens in less than an hour in most animal cells. And so you can start to think about the type of accessory proteins that might be involved with this, right? Does anyone want to shout out an accessory protein that might be involved in this process? Catenin. Yep, that's an obvious one. What's another one? Kinesin. Okay, so motor proteins are going to be involved. We'll see. Yeah. Anyone else want? Yep. Gamma tubulin, right? Because you need your gamma tubulin ring complexes to nucleate your right. Because you're going to break this down, and then you're going to duplicate your centrosome, and then you need to grow your microtubule. So you need your gamma tubulin. Yep.
How does the cell regulate? Like, so the rings is where the gamma tubulin ring complexes is where the rings come from. So like in interphase, they're coming off in every which way, but in cell division, how does the cell organize which way don't grow from that ring complex, grow from the one to this side?
So your question is, how does the cell organize the mitotic spindle into the structure of the mitotic spindle versus just random?
Yeah.
Okay. Did you have an answer?
I was going to say EV1.
EV1. Tau and MAP2. Okay. EV1 plus tips. Right? Why are you saying EV1?
Because you had a slide that said microtubule organization.
Hmm?
It said microtubule organization. That's not about all it said, but I remember EV1.
Okay, I have to think about that one a little bit more. I'm thinking more motor proteins. But let's go into the next slide because I think we start to talk. So we're going to talk a little bit about motor proteins and how motor proteins will interact with either two microtubules or microtubules and the cortex of the cell and help to kind of orient the mitotic. I want to think about EV1. Let's think about that and talk about that. It was a yeast, but they have a mitotic spindle too.
Yeah, but it said in the position. It said in the position of the mitotic.
Yeah, so that's what I'm trying to think, because it's a plus tip, right? And it's involved in positioning of the mitotic spindle, and plus tips only bind to growing microtubules, correct? So it must also have something to do with. I don't know enough. We need to talk about that. Yeah. The answer is out there. Stump the teacher. You guys want to kill me with the stump the teacher. Okay, let's do a stump the teacher because you guys love extra credit. Thank you, Mackenzie. So the question was, how is mitotic spindle organized into the mitotic spindle versus it just kind of being, microtubules growing out in every direction, like it is in the interphase centrosome. And so the answer, one of the possible answers was maybe EB1 is involved in that. And so we know it's involved in that, but the real question is how? What is the mechanism of action of EB1 in mitotic spindle assembly and organization?
Do you want specifically
EB1. I want EB1.
What if EB1 is not involved?
If it's not involved, then you say it's not involved. Yep, and so I'll put a thing on a blackboard that you can submit it through. That's worked really well with me keeping track of everything. Yes?
So the question is, what is the role of EB1 in the organization of the mitotic spindle?
Sure. What is the role of EB1 in the organization of the mitotic spindle? That was a good distillation. Yeah. Okay. Okay. Interphase microtubules undergo rare catastrophe. So they're growing and shrinking, but They undergo rare catastrophe. So what kinds of regulatory proteins are likely to be bound to interphase microtubules? This is actually a great review for the exam. What do you think is going to prevent catastrophe of microtubules in interphase? What did I hear? XMAP 215, that's very logical because it's a side binding end binding protein that stabilizes. I heard a capping protein. So maybe a protein that binds to the ends and stabilizes. What else? Those are the obvious ones, right? What proteins would you definitely not see? Stapman, because that's going to induce more catastrophe. What else? Yep. Kinesin 13, because that's a catastrophe factor. Yep. And so these proteins are all regulated, right? So how would you answer this question in the lab? So we came up, you guys just hypothesized what proteins might be interacting with interphase microtubules to prevent catastrophe. What would you do in the lab to test this hypothesis? Yeah. Okay, so you can do immunofluorescence with an antibody against these proteins, see if they co-localize with microtubules. That will tell you the protein is binding to microtubules, but will it tell you that it's preventing catastrophe? You
know the role of the protein, which you already know.
Well, you make the assumption based on the role of the protein that it's doing that. So you would do your experiment, but then you would do more experiments. Yeah.
I would knock out that gene so that I would add these proteins that cause catastrophe. See, does catastrophe occur if these proteins are knocked out?
Okay, so then you knock out the gene, which would confirm whether or not catastrophe is being caused if you get rid of those genes. Did I see your hand up? No, you were just like, okay. What else? Yep.
I'll also overexpress them, see if I get more catastrophe if they're overexpressed.
Overexpress them and see what?
See, like compared to normal levels, and see if I overexpress these genes, will there be more catastrophe?
So overexpress your genes and see if there's an effect on frequency of catastrophe.
Yeah.
But if those genes are supposed to prevent catastrophe, how do you see?
Well, what you'll have to do is see if there's any effect, right? They'll either go up, go down, or not change. Deletion should also up, down, not change, right? Okay. Okay. So motor proteins, though, do play a fairly significant role in organizing the mitotic spindle. So this is a picture I took out of a review showing your mitotic spindle and some chromosomes, and all these different motor proteins that are involved. You've got motor proteins that are minus end-directed, plus end-directed, bipolar directed, so they go in both directions because they have motor domains on either side. Then you've also got motor proteins that are going to be bound to the cortex and the microtubules. I'm not making you exactly memorize how this all works, but it kind of makes a lot of sense if you imagine. This is a very easy one to understand because you're attached to the cell cortex and you're walking along your microtubules, so that's going to help to shift your microtubules. These are easy to understand because you've got motor domains on either side and you're bound to two microtubules, and that's going to help them to slide back and forth and stay oriented the way that they're supposed to be oriented. You've got some motor proteins that are involved in interacting with chromosomes and also microtubules, so it catches those microtubules, catch those chromosomes, and then they need to get lined up. So that's a big part of the difference between your interphase microtubules and your mitotic spindle, right? So there's three classes of microtubules that make up the mitotic spindle. There are your astral microtubules, and so they are facing away from your chromosomes, and they're pushing up against the cell cortex, and they're also interacting with motor proteins in the cell cortex, and so there's pushing and pulling happening there to help to organize and bring the spindles to the poles. Then you've also got your kinetochore microtubules, and they're interacting with the kinetochores on the chromosomes. So those are the ones that are going to grab onto the chromosomes. And they also have motor proteins that are involved with orienting them. And then there are interpolar microtubules that are going to interact with the microtubules from the other pole, and there are going to be motor proteins there too, kind of helping to slide them back and forth to orient the mitotic spindle where it needs to be. Any questions about this? Okay, so that's one type of structure that needs to be built when it's needed, right? Your mitotic spindle. So another kind of structure that needs to be built when it's needed are these cell protrusions that are associated with movement. We've talked a lot about this, right? So here's your big picture slide. I pulled this off of a lab's website, actually. It's a really nice picture, though. And so what it's showing you here is you've got your pool of actin bound to profilin. And then what happens is there'll be some extracellular stimuli, right? Because signaling controls everything, right? Some kind of extracellular stimuli that's going to activate WASP. And then WASP is going to activate ARP, right? So then WASP activates ARP, and that initiates new filaments, new actin filaments, and then you get elongation of these filaments, right? Because more ARP is going to bind to the sides, and you're going to get that formation of that web on the actin. And so that's all happening. You've got your remember these are all ATP bound when they're being added, and then you're going to have ATP hydrolysis, so it's going to weaken those interactions, and you're going to get leaving from your minus end, but also you're going to have proteins that are going to be helping here. You're going to have cofilin. That's going to come and it's going to bind to your ADP bound actin and take it apart. That's your old actin, right? It's going to take it apart, and that's going to allow for formation of, well, first of all, I didn't know this actually until I read this, but profilin helps to catalyze exchange of ADP for ATP, so that recharging of your actin monomers. And so then you've got your profilin binding, and then you have more ATP actin that can then be added up at the plus end again. And so it's that pushing up against that cell cortex that's going to create those protrusions to help to move the cell forward. There's also capping proteins that will terminate elongation. So let's say the cell needs to be going in one direction, you need to control the direction that that web is being built in. So you might have some capping proteins that are going to prevent formation of a web in a different direction. And then there's also ways to regulate cofilin. Yeah.
Does formin ever act almost like synergistically with ARP? Does ARP will obviously bind at an angle, and does formin bind at the end of that actin?
So does formin, formins, the question is, does formin work with ARP because maybe formin can bind to the end of the actin and add on? The only thing though, and I'm not going to say no because I'm not an expert in cytoskeletal dynamics, but the thing to always think about when you're considering these questions is, well, what kind of structure of actin does formins make? And they make a parallel bundle that are not usually associated with the leading edge of the cell, but not to say that doesn't happen at all. It might happen in some way. It's a logical thought because you've got your plus M sitting there, right? And so why not? Why wouldn't a formin bind to it, grab profilin, that actin, and help to add on? Why not? it's very possible, because then still you'll have your sides for ARP to bind to and still make that web structure. So I didn't really answer your question because I don't know. But I like the way you're thinking. Okay, so actin, myosin, and regulatory proteins all work together, right? That's what I just showed you, right? Actually, I don't have any myosin here yet, but you're going to see it in a minute. And regulatory proteins, that's all these accessory proteins, work together to promote the cell purling, right? So you're forming this lamellipodium, and that's going to push forward. It's going to be protruding. You're going to have interaction with the substrate that the cell's walking on. And then in the rear, you have contraction. And this is where you're going to have myosin coming in and contracting inward and scooching the rear of the cell forwards. And so you're probably wondering how this happens. So we're going to talk about that. So actin polymerization at the plus end of the cortex drives membrane protrusion. And these filopodia, lamellipodia, pseudopodia that I keep talking about, they're just different kinds of membrane protrusions that are associated with motility. You guys saw this once already, right? So the ARP complex drives actin nucleation at the front of the lamellipodia, at that leading edge. So the red is actin, the green is ARP, and where you see yellow, you've got both actin and ARP. There's a huge amount of actin and ARP nucleating actin webs. And so remember that our complex is binding to the sides. You're getting this kind of two-dimensional web of actin. And so this is electron microscopy showing actually how that web of actin looks. Looks like a web of actin. And you're getting that treadmilling, right? That treadmilling that's being promoted by the activity of what? Cofillin. Right. So you get nucleation at the leading edge, and then you're going to get that depolymerization of old filaments, those ADP-bound actin, behind the leading edge by cofillin. So this is actually showing here. The red is actin, and here you're having staining of actin and cofillin, and so you can actually see you've got. The red is actin, so you've got mostly just actin here, and at the kind of right behind it, you've got cofillin acting to remove that ADP actin subunits. And so this is driving that unidirectional motion. So do you guys understand how cofillin is acting to promote that treadmilling? So it's helping with keeping that disassembly at the minus end rapid and providing enough kind of fuel in terms of now there'll be more actin subunits that can be added at the plus end. So that's that treadmilling. I almost sometimes feel like I should talk about treadmilling in this context way in the beginning because it makes so much more sense. But there's got to be a way to do it where I don't overwhelm people with concepts that are confusing, but also don't overwhelm you with huge mechanisms. It's a balance. Questions about this? So this is what it looks like. So you've got ARP binding. So basically, if you start at this end, you're going to get this net disassembly back here, right? So cofillin is going to bind and take apart the old actin, and then it can kind of diffuse to the head, to the front, to the leading edge, and then ARP can help with nucleating more actin to push the cell forward. Okay, so let's talk about the trailing edge, right? You can't just drag that. I guess you can drag the rear along, but it's helpful if you have a motor protein, myosin here, helping to contract actin there. And so here's a picture of a cell that's moving forward. Up here's the leading edge, And so what they have is actin in blue and myosin in pink, and you can see in the rear there's a lot of myosin that's interacting with that actin web to help pull the the trailing edge forward. So you're going to ask me, okay, what regulates all this? We want to learn about regulation of all this. Okay, calm down. Things are getting crazy here. We want to learn about another GTP binding protein. Give me another. So this is where I say, do you remember we learned about this in signaling? I said, I promise I'll tell you more. Cytoskeleton. So the Rho family of proteins, these are GTP binding proteins, and they're involved in actin organization, and they help to generate cell polarity in response to extracellular signals. What's cell polarity? Well, you can think of polarity in terms of the cytoskeleton. We have plus and minus, and that's two ends that are different, right? Polarity usually means two ends are different. When you talk about, and so in this case, when we talk about cell polarity, we're talking about there's your front end, your leading edge of your cell that's moving, and there's certain things that are going to be happening there that's promoting movement forward. So you've got arc, and you've got fill-in, and you've got that movement forward. And then in the rear of this cell, there's something different going on. You've got your actin web, but then there you've got myosin helping to contract and bring that rear forward. So that has to be controlled some way. How does a cell know to have two very different mechanisms going on either end of the cell? Why isn't myosin going, oops, I went to the front of the cell, oops, I'm contracting the front? Like, that doesn't happen. It's not like just this willy-nilly motor proteins just binding to cytoskeletal proteins, right? This is all orchestrated in a way to achieve function, in this case, movement in a direction, right? So that's why when I talk about polarity, I'm talking about one end of the cell is doing one thing, the other end of the cell is doing something else. There are other reasons to have polarity. Can anyone think of another example where cells have polarity? Think about tissues, right? Think about tissues and how when you have a sheet of cells, sometimes there's an apical and a basolateral sides of cells in tissues. So that's different sides that do different things. Okay. Rho. So Rho proteins are part of the RAS superfamily. So what does that tell us about the function of Rho? It's a molecular switch. Good. Usually by this time of the semester, I have people shouting out molecular switch. And so I got one. It was less of a shout, more of a whisper. A strong whisper. And I'm tired. I'm just tired. Can we just go home? Yes. Let's just do that. Let's just skip it all. Go to sleep. That's like my kids. They don't want to wake up in the morning. They're like, Mommy, I don't want to. I was like, I know. I don't want to either. But I have to. Why? This morning, my girls had a field trip this morning. And they're always like, Mommy, why can't you come on a field trip? Like, I have to work. Why do you have to work? Well, we need money. Why do we need money? We don't want to live in that tent. You know those tents under the bridge? We don't want to live there. But, this morning they're at Towson University.
Yeah, I saw some pictures from this.
Well, they're not middle school. They're in first grade. But that's good. I'm so happy because I helped set up this field trip. So one of my daughter's classes are all about, it's a place-based education focused school. So it's all about projects and getting out into the community and making connections with the community. And so they wanted to they've been making connections with two barbershops. Like, it's really cool. And then what they do is they interview these people, and so they get to work on their reading and writing through this kind of really doing something. But so the teacher was like, they're having a hip hop artist in residence at the school who's like working with them to talk about rhythm and math and cool stuff. And they're like, we want more people in the arts. And so I was like, I know someone in the dance department, and she's awesome. So I put them in touch, and they're going to go watch a college dance class, and then they're going to interview the students and the professor. And it's so awesome, because some of these kids have no one in their family who's ever been to college. And so they get to go see what that looks like. So I'm super sad, because they're there right now, and well, we should all, we should all just go over to the, just invade the dance class. That would be weird. Be like, what are you doing? You guys, who has their leotard on? Us have okay. Anyway, sorry, I'm digressing because the end of the semester, I try to keep that under control because I hear it's distracting. And so I'm trying so hard to rein it in. So, row, molecular switch, very good. That means it turns something on and off, right? Okay. And then I told you already, it also means it's a GTP binding protein. Good. So you should be thinking, oh, there's probably some gaps and all these things, it all applies, right? Okay. Right, so the RAS superfamily of proteins, they're GTP binding proteins that function as molecular switches. Just in case you forgot. Okay, so there's three of them that we're going to talk about here. RAC, Rho, and CDC42. All part of the Rho family of GTPases. They are all GTP binding proteins. They're all molecular switches, and they all turn something on and off. What do you think they're turning on and off? Huh? Actin. Something having to do with actin, right? They are turning on and off all these accessory proteins you have to learn about, and you're like, why am I learning a list of proteins and what they do? Well, a lot of it is these proteins are being regulated in order to make the cells do something. So the example here, this is from the book where you've got it's just immunofluorescent staining actin only. It's not colored, it's just white. You don't have to complain about that. Pretend it's red. In resting cells, quiescent cells, your actin staining looks like this. Nothing very exciting. But if you overexpress or inject constitutively active Rho in, you get stress fiber formation. So that Rho is turning on something that's causing formation of stress fibers. Stress fibers use parallel actin. What do you think might be involved in that? Parallel actin formants, probably. Yeah. If you express constitutively active RAC, you get this lamellipodia formation. If you express constitutively active CDC42, you get Philpodia formation. Those both are protrusions associated with motility. And so this is how we're going to connect signaling with the cytoskeleton. So how do the Rho proteins affect actin organization in response to an extracellular signal? That's the question here. So I already heard someone say, oh, maybe it's affecting hormones. Maybe it's affecting actin nucleation, right? So WASP proteins are frequently activated by CDC42 and RAC. And so if you pull up the ARP, actin nucleation by ARP-WASP pathway, then what you'll see is a receptor. What kind of receptor is that? What is this? Is this a... Yeah, thank you. GPCR. And here's an RTK, so different kinds of receptors, right, that we've learned about. And so what will happen is you get signaling through something outside the cell, binding to a receptor, all that signaling that we spoke about. And sometimes what will happen is you'll get activation of Rho. Sometimes you'll get activation of RAC. Sometimes you'll get activation of CDC42. And then you might get stress fiber formation. Then there's this protein rock that we didn't talk too much about. You'll get actin and myosin contraction. Here you have, where is it? Here you have NWASP and ARP. You guys have definitely heard about that. Activation of ARP and growth of these actin branches, that actin web. Yeah.
So when we're talking about signaling, you said that a signal could affect many different parts of the cell. So would it be also a signal to activate, like, let's say, some transcription factor and also one for.
You just answered that question on the exam. Right, so there was that question about the effector proteins. What kind of effector proteins? And I said, before the exam, I said, transcription factors, but not only that, also you can get activation out of these row family proteins that affect cytoskeleton. So that's a whole other class of effector proteins. And then another class is that apoptotic inhibitors. So proteins that affect apoptosis. But yes. And so this activated WASP binds to ARP and enhances or activates ARP nucleating activity and web formation and protrusions. So this should sound very familiar because we read a paper about this, right? Someone throw something in this direction. Yeah, we're crashing. And you know, I just got an email yesterday. Recover my work and restart. I just got, well, the good thing is we're still recording. I just got an email yesterday from OTS, and they were like, we don't need to really save that. And they were like, did you get your memory in? That'll take 2 seconds to install. And I was like, well, I believe it was not ordered yet. So annoying. I think we're okay, guys. No one needed to actually throw anything. But I wanted to. Huh? Well, I see it over here. Oh, God. It's horrible, the whole situation. Okay. You know why? Because my computer knew I was going to show you data from a paper that you read. And that might be too much for this early hour of the whole meeting. Okay. So remember we read this paper about E. coli and pedestal formation and how E. coli here recruits ARP and WASP and NIC. And then there were all of these immunofluorescence images where we're looking at EPEC and TIR and EPEC and actin. Actually, that was supposed to be alpha-actinin. EPEC and WASP. And EPEC and ARP. And then there was the NIC negative cells, which couldn't recruit ARP and WASP anymore. You need to get pedestal formation. And so I'm connecting it back so that when you guys are like, why did she make me read this paper? You can realize that there's a reason for it. Okay. So, everyone having good memories and recitation? So CDC42, when it's GTP bound, so when it's on, stabilizes the active conformation of WASP. So WASP could be in 2 different conformations. One is active, one is inactive, and only when it's active can it bind ARP and activate it. And so that's exactly what CDC42 does to encourage nucleation. So if you get activation of RAC, then you can get activation of ARP and all these, and then there's also effects on a lot of different accessory proteins, and this might look overwhelming, but I highly encourage you to open your book and look at it very closely, read the figure legend. It all makes so much sense with what you already know about these accessory proteins. So RAC is going to activate WASP, which is going to activate ARP. It also activates this other kinase, which activates filament, which is that web cross-linker. So this is all involved with this web formation. It also inhibits these this activator of myosin, MLCK, that we learned about, right? And so myosin is actually off. Makes sense. You usually don't have myosin at that leading edge of the cell. Then also you get activation of inhibition of capping proteins, right? Because you don't want capping proteins here because you want that treadmilling effect that's going to drive the cell forward. And so this is all turned on by PI3 kinase, which is activated by signaling. Right? So this can be very challenging because now you're really being forced to draw from a whole bunch of areas of the course and think about it together, but you can do it. I know you can do it. So just try to remind yourself, just think back a little bit. So Rho, another one of these GTP binding proteins, it results in nucleation of actin filaments by formins. And to go back to Austin, your question about what could formins be working with, a lot of that has to do with which GTP binding protein is activating what, and there are different proteins that are going to activate different things. Not to say that one is going to inhibit the other, but one's going to inhibit the other. So we're going to see in a minute that is how you get that polarity. So Rho is going to activate formins, and then you get that actin bundle growth, so those stress fibers are going to form. And then it's also going to activate a kinase that's going to result in inhibition of a phosphatase. So you have this myosin light chain stays phosphorylated, and you have increased myosin activity. And then it also activates a kinase that's going to keep the filament off. You want those stress fibers to be stable. And so then you get the stress fibers and focal adhesion formation, but basically formation of this contractile actin bundle. So I'm just going to take you back to this slide that I gave you guys at the end of signaling and retell you the story with all this new information you have, and hopefully you can make more sense now. Right, so we talked about signaling and how there's this F receptor, and it's involved in cell shape, right, and growth. And so with motor neurons, you only want motor neurons growing in the direction they need to grow in, and if they're going in the wrong direction, you get signaling that's going to result in collapse of the growth plane. And so that's how this is working here. When you get signaling from a receptor on an adjacent cell, it's basically saying, no, don't come here. You get activation of basically a Rho gap, and that's going to activate Rho, and then you're going to get formation of this kind of stress fibers and contractile actin. So then the whole cell is going to contract and not grow. Okay. Right. So Rho A stimulates myosin-dependent contraction of the actin cytoskeleton. So when you don't have this signal, all those Rho proteins, RAC, Rho, and CBC42, all are activated equally. And so that promotes contraction and projections and allowing growth to actually occur. But once you get the signal, it changes, and then only Rho is strongly active and the others are less active. And that promotes the contraction and the growth can collapse. Okay, how are we doing? Saturated? I'm so sorry about that. And I feel saturated myself. I hear you. Thank God for the short week next week. Okay, so another type of polarized cell growth is when you've got mating, yeast mating. And so this is an example of responding to an outside signal through a receptor and then effects on actin organization. And so when you don't have mating factor, so that's just a secreted molecule, when you don't have mating factor, you just kind of have the cells in their normal shape. And then when you do add mating factor, what happens is those signaling molecules will bind to GPCRs. Then you'll get signaling, which results in cell polarization, also called schmooing. I was getting giggles with the schmooing. And then, so then the yeast cell can mate. So this happens in times of starvation. So the yeast cell will mate and become diploid and sporulate and form spores. Don't worry about the life cycle of the yeast here because that's not what we're focusing on. What we're focusing on is the changing, that schmooing, it's changing of cell shape, and that requires actin reorganization, and that happens through CDC42. Okay, that's GPCR. And there's that. It's funny, I feel like I know this, but I don't really know this. It's like so familiar but not familiar all at the same time. Huh?
Oysters.
Oysters have schmooze?
Yeah, that's their breeds. They have like a little leg, and that's what they use.
Cool.
Yeah, that's
I feel like I have to do a lot of learning about schmooze. You know, over the break. So this is what happens, right? You get your mating factor and it binds to your GPCR, and then you get your activated G protein. And remember, G proteins activate something else, right? And so here it's going to activate CDC42, which then activates Rho formins, and then you get that actin cable formation and schmooing, and then it also activates WASP and ARB, and then you get actin polarization. And that's what needs to happen to form that schmoo. What kind of receptor is this again? Right. Just making sure. Okay, so remember we started this unit talking about movement of a white blood cell towards an infection, and motility, and all that stuff. Right, so you have chemotactic movement. So it's moving, it's chasing a bacteria because that bacteria is usually secreting something that's detected by the cell. In this case, this experiment, you're looking at a neutrophil moving towards a pipette, which you can see right here, that is leaking formal methylene leucine phenylalanine, which is bacteria. So that's the chemoattractant. That's the signaling molecule, right? And so it's saying, hey, move to me, come chase me. And so it's basically signaling, right? You've got a signaling molecule, it's going to bind to a receptor, and then what happens is you get activation of RAC, and then activation of ARP, and then movement. Okay, so that's what I was just showing you, right? Your bacteria and your chemoattractant and your receptor. And how does it really work? So what it's doing is you've got your GPCR and you've got these different G proteins that activate different things. And so what you end up having here is one G protein that will become activated and it will activate RAC. And so then you get actin polymerization and protrusion at the front of the cell only. And the reason it's limited to the front has to do with the half-life of second messengers, so they don't last too long. And then you will also get activation of a different G protein, which will activate Rho, which will be active in the rear. And that will allow for actin myosin contraction, and that's how you get that contraction in the rear. But Rho kind of inhibits RAC, and then RAC kind of inhibits Rho, so that kind of keeps those activities segregated on each side of the cell, so you get that polarized cell that's moving with different types of actin on either end. So it gets a little bit more complicated. I'm going to leave you with this. It's even more complicated, and we won't go deeply into this because I'm actually concerned that some parts of this have changed based on more recent research. But there is communication between actin and microtubules also. And so that's going to result in whole cell polarization and locomotion. So the example here is a T cell. and the target cell. So T cells are going to recognize what? Who's taking immunology? Antigen. Okay, down to what? MHC. So T cell receptors recognize some kind of peptide in the context of MHC, so another molecule on the target cell. And that interaction between a receptor and another receptor is going to cause rearrangement of actin and reorientation of the centrosome, which then reorients the Golgi. And that kind of slides the Golgi around, so you've got this immunological synapse. And that allows, so Golgi, remember, you've got secretory proteins, TR, Golgi, secretory proteins, and then they go out of the cell. And so when you have, depending on the T cell, right, in your cytotoxic T cells, you get delivery of these different molecules that will kill the target cell. And so that orients the Golgi close enough so it's very easy to kind of deliver that lethal hit to the cell that's infected that you want to kill. And I'm not going to go deep into this, but I just want you to know that I teach things in this kind of very simple, basic way, but it's not simple. I'm not saying it's easy, right? Because this is not new information. So it's challenging to learn it all, but I very much kind of try to keep it simple. But it's not. Because not only, they basically all interact with each other, and then you've got effector proteins that are downstream of this Rho family that will modulate microtubule dynamics, affecting staphin, which will then modify actin dynamics, and it's a whole area of research. Go away. I'll see some of you soon. Yes.
So one of the slides, when you incorporated the mating factor
Yes.
with the GPCR, does it still undergo so would the mating factor be the protein that kind of activates?
It's your signaling molecule.
Right.
So that's this. So if you look at this one, that's kind of that's your signaling molecule binding to your GPCR, activating your G protein, which then turns something else on. In this case, it's
The GT. Okay. So the phosphorylation would still occur with this signaling factor.
Where is
right?
the phosphorylation going to happen? So remember, with GPCRs,
There is no phosphorylation.
right.
That's
It's the
that's
activation of
the
GDP,
G got
GTP.
it,
Yep.
Okay.
Yep.
Okay. Thank you.
Yep. Oh, you're back.
Yep, I'm back. I talked to the registrar's office and they said that was fine. So I just need you to sign that, and then I got to get this filled out by whoever I'm going to transfer the credit.
Okay. Oh, I have no. Do you have a pen?
I have a pen, yeah.
Wait, where are you going?
What's the date? 20th?
20th, yeah. No, I always have to think about that.
New Zealand.
Yep.
Enjoy.
Oh, and then the second page, too, is just I have to turn it into global links so they know. The first one's for Towson, Missouri. All right, thank you. Yep. Thanks.
So, multiple choice yesterday didn't really agree.
Right. None of them. That's on purpose. Because half the class, some people asked me, and then I'm happy to tell people. I just don't want people to have access to it until everything's
Yeah,
done.
well, on the last one, I saw my score, but it didn't give me access to the questions. So I was.
Yeah,
Okay.
but then I still hit it because sometimes what happens, let's say everyone gets a 60 and the average is a 60, then I need to curve it some way because that means I did something wrong. And so I don't like to show everything till it's done because then someone's going to be like, but I had this. I thought, what happened? No one usually complains about the grade
I
going.
actually kind of like not.
Okay.
But she was saying that she saw it, and I was like, maybe that means something was wrong with my test.
Oh, no, yeah. No, you failed. That's why you couldn't see it.
You were here first.
I had a lot of questions.
Okay, I just have, yeah, mine, she kind of answered that. I was wondering when you were going to the test stuff. Because technically, do we have to, like, come or, like, know anything for the final if we are okay with our grade?
You have to know because of your love of biology.
No, I know that. I know that, but I'm just saying, so it's like I know.
Right, yes. Attendance does sometimes drop off, but if you're okay with your grade, you don't need to take the final.
Right, okay.
The tests will be up once they're graded, because I have probably 70% of the class taking it today.
Right.
Right.
Okay.
So I have to get through the grading next week sometime. The other reason to keep coming to class is because of those 5 points.
Right. No, I know that. I
Yeah.
just wanted to know.
Yeah.
You answered it.
Okay.
All right. Thank you.
Yep.
Sorry, I was looking at a couple of like, programs I wanted to apply to this summer. I think I mentioned before, like I just transferred here, so I don't really know any class that well. Would you feel comfortable writing a recommendation letter if I asked you?
I mean, I'm always happy to support my students, but you would have to give me something to go with,
Yeah.
because otherwise you're going to get, she was in my class, she seems nice.
That's what I figured, because I don't have it for like a couple months, so I wanted to ask you ahead, like maybe if I could meet with you a couple
Yeah,
times.
yeah. So let's make an appointment maybe after the semester
Okay.
is done, or at least after classes are done,
Mhm.
maybe between classes and finals or something like that. And not between, you know what I'm saying, that week of finals. And we could talk. Usually what I want from you is like your CV. If you have a personal statement you need to do, then that. Stuff like that, just so I can know something about you. Because it's always better to be like, oh, and since she was a child, she wanted to be an astrophysicist. And I don't know. Okay.
All right, thank you.
Yep. What's up?
Are we going to have recitation on Tuesday?
No, because there's no Thursday.
Okay, I just wanted to double check.
Am I still recording? Yep, I am.
You are.